Today I'm going to talk about our work with my wonderful collaborator Joel Brown on estrogen as an essential resource and the coexistence of ER-positive and ER-negative cells. Before we dive in, we need to talk about essential resources and Liebig's law of the minimum. So in 1840, Karl Sprengel noted, and then Justus von Liebig popularized the observation that it's not just the total resources that determine growth, but their ratio. And that beyond a certain point, adding more of one nutrient, such as nitrogen for plants, for example, did not increase yields because some other nutrient was the limiting resource. So he was focusing here on nitrogen and phosphorus, but really the principle applies to many cases. So with the two resources, at any given time, just one or the other resource is limiting unless they conform to a specific ratio in the diet. If you remember from chemistry, that's limiting reagents. When I think about Liebig's law of the minimum, I'm a cyclist, I think about bikes. And if you add more pedals per one frame, adding more pedals will not give you more bikes. So you have to have a proper ratio of things. That's just how we think about stuff. Essential resources impact the dynamics of all ecosystems. Could be plants or microbes, and could also be normal and cancer cells within their host. That's going to be the focus today. Okay, diving deeper into the essential resources, we can try to subdivide them into general versus specific resources. So some nutrients could be thought of as general resources, like glucose or oxygen. They are used by all cells of the body. Some nutrients can be classified as specific, like a subset of cell types or tissues need them. So we propose that hormones can actually be thought of as a specific resource in this case. Because for some cells, like in breast or in prostate, even as all the other nutrients may be in ample supply, their proliferation can be controlled by regulating this one resource, the hormones. And that makes hormones essential relative to the pool of other resources. A few words about estrogen, just in case. The ability of cells to recognize and utilize estrogen is mediated through estrogen receptors. So here I have a little diagram of what normally happens. Estrogen enters the cell, binds to the estrogen receptor, and that, once a complex is formed, that gives a signal that you can start proliferating. In cancer, once a patient's breast cancer is clinically detectable, cells of the tumor can be classified as ER positive, so they require estrogen, ER negative, so they do not require estrogen for proliferation and they lack estrogen receptors. And actually, most primary breast cancers have both types of cells coexisting. So you might have some ER positive, some ER negative coexisting in the tissue community. And they're scored as ER positive or ER negative based on the percentage of cells that actually exhibit the estrogen receptor. Actually, the cutoff, what percentage will make tumor ER positive versus ER negative is not that clear. I'm going to get to that later. But generally, patients with ER positive breast cancer have a more favorable prognosis compared to ER negative, and they have more therapeutic options. So if you end up having a breast cancer, ER positive is better. Okay, so game plan for what I'm going to be talking about today. Joel is an ecologist. We're thinking about this from an ecological point of view. And ecologically, within a breast cancer tumor, we can expect to observe at least 3 distinct types of communities. We can see all ER positive cells, or all ER negative, or some kind of form of coexistence, some kind of a mixture. And what we want to do here is to explore the circumstances that will favor ER positive versus ER negative breast cancer as a function of nutrient availability. We will model interactions of cancer cells with a general resource like glucose and a specific resource like the estrogen, subject to Liebig's law of the minimum. That's why we were talking about this earlier. We're going to look at nutrient uptake rates, resource supply rates, impact, and the subsequent impact on population composition. We'll find conditions that favor ER positive versus ER negative cancer cells in the tumor microenvironment, and then kind of discuss some strategies that may impact success, actually, of hormone-based therapeutic interventions. Okay, here we go, the model. So denote R1 to be your specific resource, pure estrogen. R2 is our general resource, glucose. And then we have 2 general types of consumers with 2 strategies. So if you're using strategy 1, you're using both specific and general resources. Those would be my ER-positive cells. They can grow faster, but are limited if there is no estrogen. So there's like a catch to this strategy. You can have consumers using strategy 2 that rely only on general resources. And they will grow slower, but they do not have resource limitations. So you have these, neither strategy is optimal, quote unquote, and it is really specific to the environment, which makes this problem interesting. Now we're going to introduce the strategy parameter alpha, such that, so if I set alpha is equal to 0, that means all of the cells in my population use glucose and are not limited by estrogen. So in previous definition, it's my second strategy, S2. If alpha is equal to 1, then the cells use both R1 and R2, so they are limited by estrogen. That's my pure strategy S1. Okay, equations. Do not be alarmed. It's not that bad. So this is my population rate of change of population of cells. So these are the ones that have alpha equals 1, so they're limited by estrogen. So x alpha equals 1 times. So this is my growth function, or the growth that is proportional to both resource 1 and resource 2. And in order to capture the limiting regions, the Liebig's law of the minimum, we're using this functional form. So it has to be a minimum of one or the other. And then you have some natural death rate. Okay, so if we have cells that just use glucose, they're not limited by R2, they have no such restrictions, so they grow really just proportionally to R2. So A's and B's and all of those things, they correspond to nutrient uptake rates and conversion of nutrients into basically biomass. It's all in the paper. It's kind of irrelevant for right now, but you, of course, can look it up. And then there's a natural death rate. And right now we're taking them having as the same natural death rate. You can relax that assumption if needed, but that's what we're doing here. Then the total population, of course, is the sum of these two subpopulations. Okay, now the resource. So R1, that's my estrogen. So there's some kind of a turnover, some kind of inflow, some outflow. So this one is only consumed by my alpha equals 1s. So by my ER-positive cells. So it's proportional to these guys. Okay, R2 is my glucose, again, some natural turnover, and this guy is consumed both by my alpha equals 1s, so the ones that are limited by estrogen, and by the other ones who are not limited by estrogen. And we wrote this in this functional form. Okay, so now we have the model, and let's ask ecologically, what do we expect to see? The outcomes of these interactions now depend on resource supply points. So what does that mean? So here's my diagram. On the x-axis here, I have the rate of inflow of my specific resource, so my estrogen. On the y-axis, I have inflow of glucose, and so what do we have? Well, we have region 0, where you have resources that are insufficient for anyone's survival. That makes sense. Death rates are greater than any of these resource consumption and conversion rates. So here we have a region when we have very little glucose and, okay, some estrogen, so we're limited by glucose. But here at least some of the cells can survive. So there's a little bit of estrogen, and the ER-positive cells are a little more efficient at converting glucose into their resources, so they can survive here with very little glucose. Okay, that's good. Now, increasing glucose, if we have very little estrogen, then kind of makes sense. Your negative cells win, outcompete everybody else. If you keep increasing the inflows of estrogen, so we get a region where you're starting to see a mixed strategy. And what's very cool here is you can actually analytically calculate the equilibrium resource levels. So a specific resource, okay, assume that estrogen, there's now more estrogen. Predictably, you are going towards more ER-positive cells. And then finally, if you have a lot of estrogen and a lot of glucose, although it's not that limited by glucose, if you have a lot of estrogen, you will have pure strategy, all ER-positive cells. Okay, so a couple of interesting points here. Even without competition, these guys, seropositive cells, will be absent if the supply points of either resource is below subsistence level. I mean, that's an important point here. And same thing, even in the absence of competition, these alpha equals 0, so they might negative cells. They'll be absent if the resource supply point for the general resource on which they depend is below its subsistence level. Basically, they do not necessarily need to compete with each other in order to eliminate each other. You can also achieve that by just resource manipulation. Okay, so just to sum up, the key outcome of this part of the analysis, so all of the qualitatively different regions shown in this figure, figure 1 in the paper, so here, can be solved analytically from the dynamics. So we very rarely actually get analytical solutions. So it was actually very exciting. It shows that it allows us to predict resource-dependent boundaries between different population compositions. So these, like the resources, these lines will determine the population composition. So this is something we potentially can play with. And specifically, we can predict resource steady-state levels given fixed population composition with respect to consumption strategy. Okay, so that was cool. Now I want to do the inverse analysis. That is, so before we had fixed population composition and we were predicting what will happen to the resources, basically finding where the resources will equilibrate given where your population is, how many ER positive versus ER negative there. Now I want to manipulate the resources and predict the composition towards which the population will evolve. To do that, I need to use a very special method called hidden keystone variable method. I'm going to very briefly go over it right now. So first, assume that each cell has a strategy alpha on an interval 0 to 1. I said 0 and 1 are pure strategies, but now I'm assuming that alpha can be anywhere in between. With this assumption, population can consist of individuals that use either the pure strategy or any mixture of the two. And then we can study the dynamics of the initial distribution of these mixed strategies through this application of our HKV method. So the derivation and everything is written up. It's in the paper. I'm not going to go over the details. It's a little mathy, but it's not that hard. If you want to dive into it in excruciating detail, you can check out our book with my dad from a few years ago. But essentially, the key point here is, so these are my equations for my resources. The key changes that after we apply the method happen. Instead of alpha, we now have expected value of alpha, which, as you can see, is now depends on time, so it can change over time. And n of t is my total population size. So I have that as opposed to x alphas. So notationally, that's really the key difference here, how to actually derive the formulas for expected value of alpha and, of course, variance of alpha, because which will change over time. Look up in the derivation. Not that hard, truly. But now we can track evolution of this population with respect to alpha. Okay, so how will this look? Let's go through this slowly. Just a reminder, so R10 is my rate of inflow of my specific resource, here estrogen. R2 is the general resource, glucose. So if alpha is equal to 0, everybody is ER negative. If alpha is equal to 1, ER positive. Okay, so let's start here. So if you have a lot of, so R10, if you have a lot of estrogen, you have here, for one, we see the total population size is the largest. So that's region 5. So region 4, sorry, right here. So everybody is ER positive. So the equilibrium value of estrogen is actually pretty high. And equilibrium value of glucose is pretty low here. And the average strategy, so expected value of alpha, is going towards ER positive cells, as we would expect. Variance goes basically very quickly to 0. Well, not very quickly, but it goes steadily to 0. So we end up with just ER positive cells here. Okay, so now let's decrease the estrogen inflow. So we're planning to be in this region here. Okay, so population size a little smaller here. Interesting. So that's smaller population size. Equilibrium level of estrogen is a little lower. but of glucose is a little higher relative to the previous case. So that's one way to distinguish between them. The average strategy is still going really towards 1, and variance is the same. So the way that you would distinguish between them, okay, so slightly small population size, slightly different relative levels of the resources at equilibrium, but in terms of population composition, we're the same. Okay, now I reduce further my estrogen inflow rate. So R10, I want to be in this region too. I expect to have a mixed strategy. So for one, population size is qualitatively smaller. So you have, okay, kind of same level of estrogens you had actually in the previous case, but much higher steady state level of glucose, okay. So what happening with the strategy? Strategy is going kind of tending towards ER negative, but not to pure. Look, it actually looks like it's equilibrating at a non-pure strategy, the mixed strategy. And what is a little even more interesting, that the variance is remaining non-zero. So you are maintaining heterogeneous population at the steady state. Huh. And then finally, I bring down my estrogen even lower, so I'm expecting to be somewhere here, and indeed, so what do we have? Even smaller population size and even smaller level of, yes, steady state level of estrogen, higher steady state level of glucose, and now we are going much more towards pure strategy, ER negative. Variance, interesting, it spikes and then goes back down. That's pretty cool. That's interesting. You can also look at how population composition changes over time, so how distribution of different clones changes over time. So, okay, the time axis is going over here, so from 0 to 10,000 time units. So where do we start? We start, so this is alpha, initial distribution of alpha, okay, so we're mostly all kind of ER positive, yeah, over here. So, and then you can see how it changes, how the population recalibrates, and then we're ending up with many more alpha equals 0 or closer to 0, and our ER positive cells just really kind of went away. But I like that you can see this recalibration of the population. Okay, so what did we observe so far? We confirmed that the model exhibits the outcomes predicted and summarized in here. So that was pretty good. When the system equilibrates, the corresponding levels of resources is as predicted by this figure. It was quite remarkable. I can basically plug it in and read off my graphs, and that's exactly what was there. When we change resource availability, we can predict if the tumor is expected to have all ER positive or ER negative or a mix of cell types. Okay, that's good. So we can modify population composition, we can influence it. And we show that populations with mixed equilibrium strategy can be heterogeneous at a steady state. Well, variance is non-zero. So, for example, like what we see sometimes in breast cancers, you have a combination of these strategies in there. Also, kind of interesting, final population size of cancer cells is actually largest when ER-positive cells dominate the tumor. So if it's like ER-positive tumors, they have better prognosis. Typically, they're more treatable, but at the same time, they are predicted to be larger. And that happens because ER-positive dominated tumors occur when ER-positive cells are limited by the general resource, but not the specific resource. And they're more efficient with using the glucose that they do have, so they can support a larger population size. Whether cancers get scored as ER-positive, that if they have a high overall density of cells and those scored as ER-negative, it's a testable prediction, so that's the final population size thing here. Would be curious if somebody knows, or if this is something that is tested up. Okay, next question. Does the initial distribution of ER-positive to ER-negative cells matter? So what I did here, run the simulations, but what I did is change the initial distribution. or rather I changed the expected value here. So I believe I took the truncated exponential initial distribution. It only has one parameter, and I would vary different parameters to see where we are starting. So like varying from the expected value of alpha, starting from very, very small to very large. And as you can see, changes in initial distribution don't actually affect the steady state values of either the expected value or the variance. So I was looking at the cases where we have non-zero variance at steady state, but only the time it takes to equilibrate. Okay. And that within the frameworks of this model, it is the relative resource supply points that will drive evolution of population over time. That means that maybe we can do some population composition steering by manipulating the resources. That's something we're going to be thinking about right now. So putting it all together, we want to have a more integrated picture of how population composition depends on resource availability and what happens with the final strategy at steady state. So this is a numerical experiment. The data were collected as follows for various combination of resource supply rates. So basically I would change my R1 zeros and I would change my R2 zeros, and I would do a 2x2 plot. And then I run the system to steady state and collect the corresponding values of population size, the resources, mean strategy, and variance, and see how they depend on like relative values of R10 versus R20, so resource supply points. Okay, so this is really kind of the main outcome of this. So set inflow specific resource on the x-axis, so of estrogen, inflow of glucose on the y-axis. First looking at population size. So as we kind of showed it before, but really confirming, it's predicted to be largest when both resources are most abundant. Okay, interesting, so that's over here. Let's look at the mean strategy at steady state. So brighter yellow, that's my alpha equals 1. So these are my ER positive cells. Okay, so specific resource, as long as you have even a little, kind of seems like even a little bit of it, you will have mostly ER positive cells. And then as you're decreasing estrogen, then we are starting to see kind of these mixture of strategies, and then this tiny sliver here when we're below subsistence level on estrogen, and so most of the cells are ER negative. Okay, now let's look at the variance. So for most of them, we do predict here to have really very heterogeneous populations, except for these mixed strategies. So mixed strategies, they coexist. There is the mixed strategy can be stable. It's not that the whole tumor is composed of just a few cells with a fixed value of alpha, but you actually maintain a distribution. You have a heterogeneous population at steady state here with respect to strategy at equilibrium. So that's kind of interesting. Okay, and here we can look at the steady states of the resources. So, okay, so they end up being kind of lower over here and kind of higher over here. This one is a little interesting. So this one was estrogen, this one was general resources. So here the pattern is actually quite different. So you have highest levels over here when you have a lot of estrogen coming in. It's because estrogen is limiting. So they are using up as much estrogen to glucose as they can, but the remaining glucose actually ends up being pretty high. And then over here, okay, so we have R2 at steady state. It's kind of general resources now kind of lower. So that's kind of interesting. That is really fully the consequence of the Liebig's law of the minimum. And then finally, what do I have here? I have mean strategy. So the expected value of alpha at steady state is on the x-axis, and I plotted total population size at steady state. And as we saw in some of the spot checks before, but now a much more comprehensive picture, yes, ER-positive tumors are predicted to be larger, at least denser, relative to ER-negative tumors. Okay, so nutrients control population composition, or at least can influence it here. So maybe this is something we can exploit. There is a concept called evolutionary steering, when you promote a more therapeutically susceptible population composition. So ideally, of course, you would steer them all to this region 0 of my figure 1, so everybody is below subsistence level. Well, unfortunately, that's not going to be possible because glucose is required by all cells of the body, so you will not be able to really drive it down below a certain level, and it is maintained at the homeostatic level. But an alternative approach, maybe we can try steering the population composition towards the ER-positive phenotype. Theoretical, not medical advice, just looking at this from a theoretical point of view. So, but in a world where we could do this, maybe having steering the composition towards the ER-positive phenotype, which is more susceptible to therapeutic interventions, would be a better idea. This is me going back to, so what exactly is the ER-positive phenotype? Turned out to be not that simple. So how to quantify the level of ER expression? Just kind of a little bit of background. Two main scoring methods, H-score and Allred score. So Allred score combines the proportion of positive staining tumor cells and the intensity of their staining, and it ranges between 0 and 8. H-score is supposed to be more comprehensive, so aims to capture full range of staining percentages and intensities in tumor samples, rather than just the average, which is the Allred score. So this one ranges from 1 to 300. Larger score corresponds to higher intensity of staining. So the higher the score, the more ER-positive you are. And a score of 1 corresponds to up to 1% of ER-positive cells among the cancer cells within the tumor. Okay, so now we know how these things are scored. Let's look at how they are classified. The question is, what fraction of ER-positive cells within the tumor is high enough to qualify the patient for hormone therapies? So what's the percentage of ER-positive cells that they need to have in their tumor to be classified as ER-positive? Turns out, not as straightforward as you'd think. So there are a couple of problems here. One is, turns out there is actually quite a bit of inconsistency between testing facilities on how they classify tumors as ER-positive versus negative. So typically, like one or several sections from a biopsy are stained for ER expression, so with immunohistochemistry, and then they look under the microscope, okay? And tumors may show a continuum of expression levels among the constituent cancer cells. Some will have no expression at all, like these would be ER negatives. But generally, all cells exhibiting expression at any intensity are reported as positive. Okay, well, apparently, even with this, there exists considerable variability between ER classifications on the same tissue block when analyzed by different labs, at least of several years ago. Maybe right now it's more digitized, maybe AI is kicking in here, but at least until very recently, this has been a problem. These discrepancies have been reported by several other authors, so that is problem number one. The second one is, assume we aligned on how we classify the proportions, the percentages, and 10 labs took the same tissue block and they all classified that, for example, you have 2% ER-positive cells. Is that good enough? Well, this question was actually addressed in 2010 at the American Society of Clinical Oncology and the College of American Pathologists joint meeting. Panel of experts agreed on setting a threshold of 1% ER-positive cancer cells for reporting a patient's breast cancer is ER-positive. And like this would give an Allred score of 3 and H-score of over 1. Well, the other problem is that this is not based on a scientific reason, but the main rationale for this cutoff is to expand access to treatment options to as many patients as possible. And that is definitely 1,000% an important consideration, but does it actually correspond to efficacy? Well, so Morgan et al. Show that at lower levels of expression with H-score under 50, they had a lower overall disease-free survival when treated only with endocrine therapy. Chan et al. Showed that patients with ER-positive staining between 1 and 9% gained no significant benefit from endocrine therapy, unlike tumors with over 10% positive staining. Raghav et al. Showed that patients with tumors with 1 to 5% ER-positive expression gained no clinical benefit from endocrine therapy. So, I mean, obviously it seems that the cutoff is too low. I'm not going to go into discussion of accessibility to it. People should have access to therapies, but those therapies should be effective. So the question could be here then, well, yes, if therapeutic success would be greater if there were more ER-positive cells in the tumor, can we do something about it? Can we do something like resource-mediated evolutionary steering? So as we have seen above, the proportion of ER-positive cells that rely on both resources increases with increased estrogen inflow or with reduced glucose inflow. Maybe there's, I mean, again, not medical advice, talking completely theoretically just as a scientist, but in a world where this could be done, externally increasing estrogen concentration to favor ER-positive cells over ER-negative without, if this were possible to do it without compromising patient health, that would be feasible. So that would be a therapy that would be called evolutionary gambit or sucker's gambit, funny name. Some studies have showed that glucose deprivation may be able to achieve this effect, but of course, blood glucose is said to tightly regulated below 100 milligrams per deciliter, I think. Not going to be able to really drop it. So glucose is not going to be a very good thing to manipulate unless, of course, we're talking about high blood glucose and that. Well, so let's maybe kind of theoretically talk about the impact of resource-dependent intervention strategies on predicted tumor composition. Like, remember my diagram here. In a world where you were able to do glucose deprivation, like normalized blood glucose, if it's high, always a good thing. Well, theoretically, maybe you would be able to steer it towards ER-positive. Estrogen deprivation will definitely steer it towards ER-negative. So if you started with that, you would definitely at least prime your tumor to be primarily ER-negative and now not responsive to any of the hormonal therapies. So it may be in some world where you can maybe not do estrogen deprivation, maybe normalize blood sugar, maybe do some kind of manipulation here, basically favor ER-positive cells, which as we've shown, you can do through resource manipulation. You sensitize this population to get them at least to 10% ER-positive cells, as at least some of those studies were showing. Repeating once again, not a medical doctor, this is theory, but still theoretically. Then priming the tumor like this and then applying hormone therapy might be able to have better outcomes. This is the paper. It came out a couple of years ago. As always, let me know if it makes sense. If you have experiments or data, I love experiments or data, or like some data disproving these ideas. I'm happy to discuss why. Absolutely, falsified hypotheses. If you want to chat science, please reach out. And thank you for your attention.